Cell replication. To understand cell replication, we are going into the details of mitosis and meiosis in this module. This module is based upon the mitosis, and the next will be on the meiosis for the understanding of cell replication. Mitosis is one of the two types of divisions through which cell divides. Mitosis. Usually, somatic cells, there are two types of cells in the body of an organism. Some cells are called somatic cells, and some other cells are called germ cells. In somatic cells specifically, mitosis takes place to increase the number of cells in the body. So we can say that this is basically taking place for the sake of growth of the organism. We also know, if we refresh our basic concepts, previous concepts, that new cells arise in the body either for the purpose of growth, or to replace the older cells, or to replace the damaged cells, because wherever there will be a damage to the body or there are injuries to the body, mitosis takes place over there to replace these damaged types of cells or old types of cells from that specific location. There are some tissues in the body where mitosis doesn't take place, but usually in the majority of the cells, mitosis do occur over there. So basically it's for the growth, but also for the replacing of the older and damaged cells. The basic concept of the mitosis is that, in very simple words, we can say that it provides a copy of the cells with the same amount of DNA, with the same copy of DNA in the new cells as compared to the parental cells. So if a cell is going to divide through mitosis, it will produce two daughter cells. One parent cell will result in two daughter cells, and each daughter cell will receive the same number and quantity of DNA or the chromosome as was present in the parental cell, in the mother cell. The instruction making the cell is coded in the DNA. So the DNA must be replicated before the division of the cell through the process of mitosis. So there must be a faithful process of replication present in the cell, and the cells do have a very much faithful replication mechanism in them. They replicate very faithfully, usually without errors. Errors do occur, but the probability of these errors in replication is very, very low. By errors, I mean different types of mutation, which spontaneous mutation, which do occur during the process of replication, may either be insertion, deletions, or substitution, or whatever they are. We are not going into their details, but usually the process of replication is a very faithful process, and to that replication process, the new cells receives the intact copy, the same copy of the parental DNA. So DNA must be replicated before the process of division. And each new cell will have an identical copy of the same DNA as like the parental DNA, because if they do not receive the same copy of the parental DNA as in the parental DNA, then the new cell will be totally different from the parental DNA. So we have already been discussed it that characters are being passed from the parents to offspring through this genetic material. So that's why the parents are transferring the same characters to the offspring, though with little variation with some other aspects which we will discuss in recombination or in meiosis, but the characters do remain the same. The offsprings remain the same as like their parents, majorly, with some variations, but majorly they remain the same. For example, a human will produce a human baby, or doggy will produce a doggy like his or herself. So the species integrity remains the same due to this faithful replication process or division process. Now we can sum up this mitosis to divide this. The mitosis process can be divided into four main phases. They are prophase, metaphase, anaphase, and telophase. In prophase, the nuclear membrane and the nucleolus of the cell disintegrates and broken down, and so they disintegrates and disappears, and the genetic material are now in direct contact in the cytoplasm. Chromosomes continue condensing, and then now during the prophase they will be visible in the cell as compact bodies. Before prophase they were not compact, so were not easily visible as the chromosomal structures in the cell. And spindle fibers called kinetochores, they get attached to the kinetochore, or the centromere of the chromosome, the point of attachment of the two chromatids, the two arms of the chromosome. They get spindles get attached to these centromeres, and they will make a pull, exert a pull on these centromere to get it apart in the next phase. Metaphase is the next phase of the prophase. The chromosomes are now arranged in a systematic manner to the spindle fibers through the kinetochores, the kinetochore fibers, and now it's exerting pull on these centromere. But in this phase, the chromosomes are very much aligned at the equator of the cell. They are aligned at the equator of the cell and are attached to the kinetochore fibers. So the chromosomes are now lined up at the equator. Here we can see that these are the kinetochores fibers, and the chromosomes are lined faithfully on the equator of the cell. These are basically the entities, the esters, which are radiating these kinetochore fibers towards the centromere of the chromosome. Anaphase is the third phase of the mitotic division. It occurs rapidly, so have very short time span for this phase. Sister chromatids are pulled apart by the heavy pull exerted by the esters, by the kinetochore fibers towards the opposite poles, and now they are separated from each other and started moving towards the opposite poles. Here we can see in this diagram that these kinetochore fibers are exerting pull, and now the chromatids are now get apart from each other, and they are moving toward this pole. It's moving this way, and these are moving this way toward this pole, and now they are moving gradually and will be grouped over here and here in the next phase of the telophase. And telophase, the sister chromatids, they reach to their respective poles. The spindle fibers will now disassemble and will disappear. The nuclear envelope and the nuclear membrane, they reappear in the cell. The nucleolus reappears, and then division of the cytoplasm will take place, and the cell will divide into two daughter cells with the same number of chromosomes in each of the two cells. Now there is an animation which will highlight, which we discussed about the mitotic division in this module. Now the chromosomes are compacting, they are visible, the nuclear membrane disappeared, the chromosome are arranging on the equator, the kinetochore are attaching, they are pulled apart to the opposite poles, and now nuclear membrane reappear, and division of the cytoplasm then takes place, and this is the whole of the cell division in this way.